I'm very glad to be here at this fourth International Conference on Vaccines and Vaccinology. There's no question that vaccines and vaccinology over the past couple hundred years have done an enormous job in saving lives. In fact, in the last 50 years, I would say that it's had more benefit to human health than about any other medical endeavor that's been carried on. In terms of vaccines and vaccinology, I would say that more lives have been saved throughout the world by the development of vaccines over the last 50 years. And because of that, there has been one major failure of vaccines in research, and that's been in the area of cancer research. Somewhere along the line, it was decided that we can make vaccines that will cure tumors, and unlike other disciplines where we have developed successful vaccines, we launched into this area without knowing enough of the biology of the disease, cancer, in fact, totally misjudged it, and did not go through the disciplined steps of identifying the patient population that should be treated, the prevention versus therapy approach to the vaccine, and failed miserably. Of all the vaccines put into clinical trials, phase 3 clinical trials, I would say, over 80% of them have failed. Only one has been approved, and that company is going bankrupt and is up for sale now because of the unsuccessful nature of their product. Not so much the fact that it doesn't work and prolong survival for about 4 months, but the cost per life year gained is about $273,000, which is absolutely impossible. So I want to give you a little background in this area and tell you a way out of it, I think. I think I'm supposed to touch one of these. I had the good fortune while at the National Cancer Institute to have an animal model which consisted of inbred guinea pigs. These strain 2 guinea pigs had a progressor tumor, transplantable progressor tumor, which is called line 10, and it grew and killed the animals in about 90 days. The interest in vaccines I developed from having watched the original work that was done in these guinea pigs, which is bone marrow transplantation. Egon Lorenz and Charles Congdon at the NIH started studying bone marrow transplantation in this animal model, and however, when we went from this animal model to mice and to man, we got what's called secondary disease, graft versus host response. And when I went to Oak Ridge as a graduate student, working with Charles Congdon, the first thing he told me to do was to look at this in the microscope, and what I saw was this enormous immune response. And he said, we got to figure out what this is, and my thought was, boy, if we can get this to do the same thing against a tumor, it would be magnificent. And so we did identify it as graft versus host disease, and I decided I was going to spend the rest of my career trying to get this immune response to attack cancer in the same way it attacked self in the situation. So I started studying this disease in the animal model, and the guinea pigs were raised at the National Cancer Institute's facility in Frederick, Maryland. And for a period of time, I had control of it. So I was able to use it, the guinea pigs, for my studies. And what we did was we started to artificially transplant this line 10 hepatocarcinoma intravenously into the guinea pig and watched the ability of vaccinations to effectively cure or prevent the establishment of fatal disease. And what we discovered was, if you look at the lungs of these guinea pigs after an intravenous injection of either 10 to the 5 or 10 to the 6 tumor cells, day 1 after injection, you saw what is basically the origin of cancer, one cell or a couple of cells, rogue cells dividing in an unregulated fashion in the lung. And by day 4, these cells have formed a small cluster. And by day 7 to 10, they're about 0.1 to 0.2 millimeters in size. And all of this is microscopic only. It is not visible. It's occult disease at this stage. But if you go out here to day 21, you see a formation of a tumor nodule. And this is barely visible to the naked eye in the lungs at this time, but I'll show it to you microscopically. And what we did was we started vaccinating day 1, day 4, day 7, day 10, and followed the survival of these guinea pigs. We used BCG as our adjuvant. I know you have a whole session on adjuvants, and I know the last one you would pick is BCG, but I did pick it because we had done a big study on adjuvants for polyvalent cellular vaccines, and we found this was the one that worked. All the other ones didn't work, didn't make a dent in it. And I'll show you why BCG alone doesn't work and that it doesn't work in this model. And what we saw was, first of all, this is the nodule that we see at 21 to 24 days. This is an established metastatic nodule. It has the peripheral circulation that one sees in these nodules. That is, the blood vessels form in the circumference around the tumor, such that in the center of the tumor there's very little circulation and it's actually hypoxic. I believe at this stage of tumor formation is where the contact inhibitors of the immune response actually start to express themselves. So that basically today what we know is that at this stage of the tumor, which is barely macroscopic, there are PD-1 and PD-L1 contact inhibitors that when the lymphocytes, the cytotoxic lymphocytes, enter into this area to mount a response against a tumor, they're turned into exhausted phenotypes by these two proteins. There are others besides PD-L1 and PD-1, but at this point, vaccines have an uphill battle. They have to create an immune response that could get around the contact inhibitors in established tumors. So when we looked at the survival of these guinea pigs being challenged with the hepatocarcinoma and then vaccinated, the small occult disease patients, guinea pigs rather, showed about 66 to 73% survival. If we go out to day 4 and 7, you see about 40% survival. And if you go out to day 20 to 21, where I showed you the size of the nodule, you get improved survival-free interval, but eventually all the animals will die in spite of the vaccinations. So we knew this at that point, that a vaccine should be used to treat and prevent occult disease from establishing, not used to treat established disease. Not just because of the contact inhibitors that may be present, but because there is not enough immunity to do the job. When we looked at this data, again, comparing it to the controls, BCG alone at either 10 to the 5 or 10 to the 6 IV injections to establish metastatic disease had no benefit. The tumor cells alone showed no benefit at either intravenous dose. BCG in line 10 at 10 to the 7 or 10 to the 8 showed some benefit in combination admixing. But then as we improved the combination to 2 injections or 2 vaccinations, we started to get better effect. And at 10 to the 7 or 10 to the 8 BCG, with 10 to the 7 live metabolically active line 10 cells, 2 vaccinations a week apart, we got cures at both doses. So it tells us that this can be done. You can cure occult disease, but you have to optimize your regimen and you have to have the proper balance of the adjuvant and the tumor cells. And what we learned in biotherapy that is not true in chemotherapy is more is not always better. In other words, the more adjuvant you give, if you increase the dose, this is a C-parvum or BCG as an adjuvant, at 7 micrograms of C-parvum or 10 to the 6 BCG, you get no clinical benefit. At 70 and 10 to the 7 BCG, you get clinical benefit in both of two experiments. Then when you go higher in BCG to 10 to the 8 or 700 micrograms of C-parvum, you start to see the benefit decrease, which is what we've seen in this situation and which we also saw when we tried to use BCG as a curative for bladder cancer, which you might know right now it's approved as it's been approved since the early 90s as a therapy of choice for carcinoma in situ of the bladder. This is a pre-metastatic bladder tumor. And it was discovered that more BCG is not better, it's actually harmful. So again, the biotherapy rule, unlike chemotherapy, is more is not better. In fact, you get this bell-shaped curve that means you've got to optimize the regimen. So in other words, what we're saying is if you look at the profile of cancer, it takes between 1 and 10% of a person's lifetime for the tumor to start where I showed you was the original few cells in and around an arteriole in the lung, to an established tumor of 10 to the 10 detectable size, detectable by whatever manner you choose to use, at which point we do surgery, radiation, and chemotherapy, and we get decrease in the volume of the tumor. Depending on how successful you are, you'll get greater or lesser reduction of the tumor, which will then recur, and patients will die from recurrence. If you treat at the optimum time when you have minimal residual disease, as we saw in the guinea pigs, you actually can get cures. So basically what happens is you have to learn to use the vaccine. You have to have a vaccine that gives you a robust immune response, and you use it to prevent recurrence. 80% of the patients that die from cancer today die from those occult-celled recurrences. What did the industry do, you might ask? Well, the industry, which was made up of probably very few immunologists, very abundant number of oncologists who had experience with chemotherapy, decided to treat advanced disease. Why did they treat advanced disease? Because they had to raise money to do these expensive phase 3 trials, and the investors wanted 3-year return on their investment. The big pharmaceutical companies didn't want to do it this way because it doesn't fit their paradigm. It's too much work and too much effort to save lives. But what the industry did was companies started up, and some established companies, and they treated late-stage disease with either autologous dendritic cells or late-stage disease with allogeneic off-the-shelf antigens. And and over 77% of these have failed, and now we can add a few more failures to the list. Glaxo's MODS-3 has been declared a failure by Glaxo. They're not going to proceed. And another one by Merck, that one has also been pulled off the shelf. It's also a failure. So that's what's happened to the industry. We, having had the experience with the guinea pigs, treated occult disease with autologous patient-specific tumor cells. And I'll explain why autologous patient-specific tumor cells. And that's because in the last 7 years, what we've learned about cancer is that there's now indisputable evidence for the fact that the cancer genome, the DNA sequencing data has shown an extreme genetic diversity. In other words, cancer is a heterogeneous disease. We were led to believe by the early work of Peter Knoll that there were mutations in cancers, but 1 or 2 clones of cells mutated clones survived and formed the cancer. And that's not true. That never was true. If there are 80 mutations in the evolution of a tumor, you have 80 different mutations with 80 different protein products. If you have, in the case of melanoma, 200 mutations in an established tumor, you have all of those protein products. If you're going to form a vaccine, you need to use all the antigens. And that's not what we saw in the industry. First of all, I got to tell you that a friend of mine who worked with me at the NCI at the Cancer Research Facility in Frederick, Maryland, described in 1977 that cancer metastasis is heterogeneous. And that was my friend Josh Fidler, who together with Margaret Kripke published, 1977, I can hardly remember back that far, published a paper in Science that showed that metastasis resulted from pre-existent variant cells within a malignant tumor. And I don't think there's probably too many people in this room who haven't worked with the B16 melanoma or the B12 melanoma or the B5 melanoma. Those were the variants. B16 was the most metastatic, and everything below it was less metastatic, to the point where some of the lines didn't metastasize at all. So we knew in '77 that there was a good chance that cancer was not homogeneous, that it was heterogeneous. And at that point, I was beginning my antigen discovery part of my vaccine development. And I had no choice but to assume if there's phenotypic heterogeneity for metastasis, there very well could be antigenic heterogeneity. So we decided to go with autologous tumors in humans with colon cancer. And when we went to the we started the work at Johns Hopkins in Baltimore, Maryland. And when I met with the Johns Hopkins IRB committee, they said, "You're going to put those tumor cells where?" And I said, "We're going to inject them intradermally." And they said, "Well, how do you know they're not going to grow into tumors?" I said, "We radiate them with 2,000 rads. And that makes them non-tumorigenic, but they're still live and metabolically active." And they said, "We'll approve it, but you got to increase the radiation to 20,000 rads." So I did, and we started our clinical trials at Johns Hopkins. Today, my good friend Bert Vogelsteins at Johns Hopkins pointed out that this heterogeneity is not just within metastatic phenotypes, but the heterogeneity is to the extent that tumors of two different patients differ in terms of their mutations. So that there's diversity between patients, and you have this intra-tumoral heterogeneity, and he showed that with landscapes of the various mutations and their protein products. And here are the landscapes of two different colon cancers. He did it in over 1,000 of them. And the yellow are the mutation and mutational products of one tumor, and the white are the mutational products of another tumor. These are in colon cancers where there's an average of 80 mutations per established tumor, and only 3 of them were common. So it's a little bit like the influenza story. If you don't have all the serotypes, you don't get good protection. And when you only have 3 out of 80, I can assure you, you're not going to vaccinate anybody against cancer. And then another study, this was within the last 5 years, showed that there's heterogeneity, intra-tumoral heterogeneity within a tumor. And that was a study by Yoshida and Staunton recently in Great Britain with renal cancer, showed that if you segment the tumor and analyze each segment, you get clones of different mutations distributed throughout the tumor. So I ask you, how do you expect to do patient-specific therapy when all you have is a biopsy? And how do you expect to develop drug-sensitive, drugs that are effective to a particular single antigen when you have this much diversity? It's like saying, I'm going to win the war with a handful of snipers. It just doesn't work. So the basic tenet of this presentation is we cannot treat a heterogeneous disease with a homogeneous treatment. So a single protein product or a single class of allogeneic or even syngeneic cells won't work on all patients. And as immunologists, we're aware of one modality that exists that can address this magnitude of cancer diversity, and that's the immune system. So we pursued the development of an autologous tumor cell vaccine for each patient, and the product is called OncoVax. It's a patient-specific immunotherapy using their tumor cells, live metabolically active non-tumorigenic tumor cells, as an immune stimulant together with BCG. It induces a functional anti-tumor immune response. The immunotherapy delays the time of recurrence, and the concept is based on the recognition of tumor heterogeneity, that the genetic mutations in cancer cells may vary in every patient. So we have no preconception of known or shared antigens because it's problematic to do that. We developed a procedure, optimize our regimen. So the patients go to surgery, the surgeon removes the tumor, it's properly dissected according to our SOP, shipped as a transplant shipment to our manufacturing facility, and 6 hours later after it's received there, the vaccine is made and the quality assurance and quality control release is underway. It takes 21 days to get the sterility data back. So we have it for about a month, the patient doesn't get treated. And then we start treating. The first injection is 10 to the 7 tumor cells plus 10 to the 7 BCG. The second injection is 10 to the 7 tumor cells plus 10 to the 7 BCG. The third tumor is irradiated tumor cells alone. And the boost 6 months later is the same. In those two treatments, we get the one test that everybody recognizes the most relevant in vivo test for T-cell immunity, and that's the Latetypehypersensitivity. We get nodules between 1 to 2 centimeters in diameter. And when we get those at the time of the third vaccination, we know we haven't been shooting blanks. And when we get it at the boost, we know for sure that patients now got immunologic memory and has enough robust immunity to deal with occult disease. This is what I'm describing to you, is the Latetypehypersensitivity. It occurs within 6 weeks after we've started treatment. And then if you don't do anything, if you end at, like the guinea pig told us, only give 3 injections, it wanes with time. So this is why we boost at 6 months. It comes right back up and stays up. And this is the delayed type hypersensitivity that we measure within an erythema, which occurs against, this is antibody against the bacterial flora of the tumor. The nodules are between 1.5 and 2 centimeters. When the FDA came back and told us we had to have a sterile product, drug product, we managed with the use of antibiotics, which they permitted, to get the microflora eliminated. And what happened then was this erythema reduced by 7-fold. But the indurations didn't change a millimeter. So we got rid of the flora, we made a microflora, we made a sterile vaccine, and we had delayed type hypersensitivity that was equivalent to the non-sterile vaccine. And that is the product that we're now taking to our second phase 3B study. And to show you how important delayed type hypersensitivity is, we did a previous study with the Eastern Cooperative Oncology Group where we didn't have a sterile product, where we didn't have the kind of deliberate manufacturing procedures that we have been taught by the FDA. And we found, and the only reason this study was published was because of this data, if the patients had a significant delayed type hypersensitivity to their vaccinations as compared to those that did not, there was a statistically significant difference in 5-year survival and 5-year disease-free survival. So this DTH of the skin to the third and fourth, the boost, is indicative of clinical benefit. It ties 100% to clinical benefit. So now what we do is we boost the patients to get memory and longer T-cell immunity, and we get DTH to both of those. And that's the basis of our new phase 3 study with the sterile product. This is the data of the previous study with the non-sterile product, and we show a significant difference in recurrence-free survival, and that is now the primary endpoint of our new study, not survival alone. Survival alone is the, well, survival, I want to go back. How do I go back? How do I go back with this?
Page up. Page up. Page up. The arrow pointing upward.
Oh. I'm sorry. I didn't. That's going forward. I want to go back. Keep going. One more. Oh. Well, I was going to show you the survival benefit, but I can't because for some reason that's not on the slide. But I will go to the other slide, which I think is even more important, and that is the survival benefit I was going to show you was 5 years survival benefit, overall survival benefit. But last year, one of the medical doctors at the Free University where this study was performed went back and reviewed all the slides again and was looking for two markers, microsatellite stability and microsatellite instability. And what he discovered was that a 15-year follow-up of this population of patients still had significant survival benefit, as you see in this slide. So not only do you get an immediate clinical benefit, but it lasts for 15 years. And that's the kind of response treatment in cancer patients that you'd like to see. And so we're repeating this study. We have just raised a little over $80 million to do this new phase 3 trial, which was not an easy task, but we were successful. And I want to show you some of the pharmacoeconomic benefits of this study to our only comparable. Am I okay? Oh, you're right. I have 5 more minutes because I didn't take my 5. So, darn. I want to show you the pharmacoeconomics because one of the important things about vaccines is, and we've seen this in the past, that there are some of these vaccines that we've made that in the world we can't even afford to use because the drug companies won't make enough money selling them for the money they put into it. Well, our only comparable is Provenge, the prostate cancer vaccine made by Dendreon. And looking at their life year gain, cost per life year gained, and their margins, it shows you why the business is not going to make it. In our case, if we charge $50,000 to $60,000 per treatment, based on the 3-year survival that I was going to show you but couldn't, the cost per life year gain would be about $50,000. Now, adding the 15-year benefit that we see, the cost per life year gain is going to be about $12,000. This was recently recalculated by a group of physicians at Columbia Presbyterian Hospital. Our margin's about 85%. So from an economic point of view, the delivery of the drug is possible. The vaccinations, you have an early indication of the fact that your vaccines are working, and you get clinical benefit at a magnitude not previously seen with other chemotherapeutic drugs. So our new study, which we're launching now, while I was gone, they started bringing in sites. is a phase 3b study in stage 2 colon cancer patients and a phase 1-2 study in phase 3 colon cancer patients. In this case, it's Oncovax alone compared to surgery alone, which is standard of care. In this case, it's Oncovax plus Folfox compared to Folfox alone, which is standard of care. And we were given from the FDA a special protocol assessment and a fast track designation, which allows us to do an interim analysis in 2016 once 66% of the expected events occur. And at that point, we'll be able to file for our new drug application. So basically what we've been able to do is, I've just told you about Oncovax, which is taking the primary tumor, dissociating the tumor, irradiating the cells, and mixing with BCG and immunizing the patients back for clinical benefit in treating occult disease, protecting them from later metastasis and mortality. And we also found, we published and have patented some of them, that we could take white blood cells during a window of opportunity in these patients and get B cells that are making human monoclonal antibodies specific to tumor antigens. And we've used this already to develop a diagnostic, which was approved by the EMA, but we haven't commercialized it yet. And we're using it to probe for tumor antigens with the possibility of coming up with an epitope-based vaccine. I have to show you this because the lawyers tell me to, but I'm not going to read it. Thank you.
Comment on a great presentation. Can you comment on the decision to administer the cells intradermally versus another route, sub-Q or IM?
Yeah, that's a good question. I wish I'd have thought to give you the answer during my presentation. In the dermis, there are cells which we always call Langerhans cells. That is the greatest source of what we now call antigen-presenting dendritic cells. So if you want the antigens carried on dendritic cells, you give intradermal injections. If you want to bypass the dendritic cells, then you give subcutaneous. It's an additional obstacle because what we find is that not many people can effectively give a 0.2 of an ml intradermal injection. And that's one of the things we have to train the sites to do and not lose vaccine in doing it. And it's very doable, but I'm glad you asked that question. We don't have to have a dendritic cell-based vaccine. Nature provides it to us through the intradermal injection. Very astute of you, thank you.
So thanks, Michael, and I've just brought them in, but here's your certificate for your presentation.
I couldn't be more pleased.
I thought you